Hi, in this video we're going to talk about regulation of gene expression. Gene expression is just simply protein synthesis, transcription, and translation. So in this video we're going to talk about regulating gene expression of a prokaryotic organism. So this structure that we're going to be talking about is called an operon. An operon is a group of structural and regulatory genes that function as a single group, and it includes several parts. The first part is the actual regulator gene. Genes code for proteins, and this particular gene codes for a repressor protein. The repressor protein controls, it's a protein that controls if the operon is active or if it is inactive. And the regulator gene is actually located outside of the operon. You can see down below we've got the regulator gene right here. And the operon is all of this. So the operon includes the promoter. The promoter is a sequence of DNA where the RNA polymerase will attach. If you remember in transcription, RNA polymerase will attach to the DNA and begin transcription. So that happens at the promoter. There's also something called an operator. This is a short sequence of DNA where an active repressor can bind. So you can see that piece down here at the bottom. This is an active repressor, and it is actually bound right here. The repressor actually binds to the operator, and it will actually prevent the RNA polymerase from attaching to the promoter. If it cannot attach to the promoter here, then the RNA polymerase cannot proceed down to where the gene is. So the structural genes, those are, they come after the operator. So first we have the promoter, then we have the operator, then we have the genes. And notice it is plural. So the structural genes are one or more genes that code for enzymes of a metabolic pathway. And they are transcribed as one unit. So if we transcribe one of those genes, we transcribe them all. So we're going to talk first about a repressible operon. A trip operon, and it's abbreviated TRP, and yes, it is in italics. The trip operon is an operon that actually synthesizes the amino acid tryptophan. So this is, the trip operon is an example of a repressible operon, and you'll understand why it's repressible in just a moment. This operon exists in the on condition or the on position. So in other words, when tryptophan is absent, RNA polymerase can bind to the promoter. So we're in a situation where we need tryptophan. So we have a situation where this repressor is inactive, and you can see this binding site right here does not match the active site, so it cannot bind. So it cannot stop the RNA polymerase from proceeding past the operon down to the structural genes and doing translation. So this is in an on condition. So E. coli produces 5 enzymes as part of the anabolic pathway to make tryptophan. Anabolic pathways are pathways that build things up, that make things. So if tryptophan is already present, these enzymes are not needed. So if we have tryptophan, we don't need enzymes that make tryptophan. And the operon is turned off. This is called feedback inhibition. Now, the way this actually happens is tryptophan will bind to the inactive receptor, and you can see that the active site right here is rounded. Well, once the tryptophan binds, you can see that it actually changes that active site on this inactive repressor and actually makes it active. So this complex will then attach to this location right here on the DNA and will actually stop the RNA polymerase from being able to bind and proceed down the process to the actual genes. So the repressor is actually an allosteric protein. So if you remember, an allosteric protein is a protein that has a binding site on both sides. In this case, we've got a binding site on this side where the tryptophan binds, and on this side where it actually binds to the DNA. So an allosteric site has two binding sites. It has two active sites, one on each end. So one side is for the tryptophan, the other side is for the operator. So tryptophan is the co-repressor in this case because it actually binds to the inactive repressor, activating it. It changes the shape of the repressor. And now it can bond to the operator, blocking the RNA polymerase from doing transcription. So as the tryptophan is used up, so if we were to use up this tryptophan is no longer available. Now this tryptophan is no longer available. If we pull this tryptophan out of this receptor site, what's going to happen is this active site here is going to revert back to what it looked like here. It's going to revert back to creating an inactive repressor. So that repressor will then fall out of the DNA and will no longer be stopping transcription. So usually these types of systems are found in anabolic pathways that synthesize substances that are needed by the cell. So anabolic means to build up. So we're actually making substances. In this case, we would be making tryptophan, and this is a repressible operon. So to summarize a repressible operon, an example would be the TRIP operon. That's the model that you're going to use for any repressible operon in a prokaryotic cell. It exists in an on or a go condition. So because of that, the natural state of the repressor is inactive. So the repressor is not plugged in. So it is allowing transcription to go on all the time. And this is found in anabolic pathways. Which is biosynthesis. So prokaryotic regulation, we also have what are called inducible operons. Inducible operons work very much the same way, but are opposite. So the example we're going to talk about for this one is called a lac operon. If E. coli, which is a prokaryotic organism, if it's denied glucose, glucose is its source of food and food for its energy, and it's given lactose instead. Lactose is made up, it's a polymer, it's made up of glucose and another sugar molecule. The E. coli is interested in the glucose, so what it has to do is it has to break apart the lactose. So what it does is it makes 3 enzymes that are encoded for by 3 genes to metabolize the lactose to be able to get at the glucose. So these 3 genes are adjacent to each other on the chromosome, and they're under the control of one promoter and one operator. So they are all part of one operon, and that is an inducible operon. So prokaryotic regulation, inducible operons. You can see here we've got an inducible operon. Just like before, we have a promoter, we have an operator, we have a repressor, and we have a regulator gene. So the regulator gene is responsible for making the repressor. And so let's talk about an inducible operon. In the absence, this is active in the absence of lactose. So the regulator gene codes for a lac operon repressor protein, and it binds to the operator. So the repressor is in position normally. So it's repressing, it's stopping transcription from taking place. You can see the RNA polymerase cannot bind around the promoter because the repressor is plugged into the operator. So this is naturally in the stop position. So in the presence of lactose, lactose will bind to the repressor. So you can see we've got the active repressor here. And what happens is lactose actually fits into this binding site right here, and it will, when it's active, you can see the other side of this allosteric protein looks like this. When it binds, you can see excuse me, that second binding site has changed, which has caused this now inactive repressor to fall out of the operator. It can no longer stop the process of transcription. So RNA polymerase is able to bind to the promoter, it's able to pass by the operator, and transcription can take place. And as you can see, mRNA is being made for each of those 3 enzymes that need to be produced in order to break down the lactase. So we have something called an inducer. An inducer is any substance, which in this case is lactose, that can bind to a particular repressor protein, preventing the repressor from binding to a particular operator. Consequently, RNA polymerase can bind to the promoter and transcribe the structural gene. So an inducer in this case would be the lactose. This is an example of an inducible operon because it's in the off position, but we can force it to turn on. That's why we call it an inducible operon. It's usually found in catabolic pathways that break down nutrients. To catabolize something means to break it down. And these are generally only active when nutrients are present because that's what this organism would be trying to break down. So inducible operon summary is this, and the example that you've been given is the lac operon. It exists in the off or the stop condition. So since it's in the off condition, the nature of the repressor is active. So when this is in a natural position, the repressor will be in the binding site and will be stopping the operon from operating. But we can pull it out. That's why it's inducible. These are found in catabolic pathways. In other words, these are found in pathways where we break down molecules. Now, both repressible and inducible operons, these are both types of negative gene regulation. So what we're doing in this case is we're saying don't. That's why it's called a negative gene regulation. We also have something called positive gene regulation. And again, we're still talking about prokaryotic regulation. So this picture that you see down here, this is cyclic AMP. So I'm sure you've heard of ATP and ADP. Well, this is AMP. It's adenosine monophosphate. There's one phosphate group, and what happens is that phosphate group will actually bend back on itself. It'll bend back and bind in 2 places, both here and here, to the ribose. So E. coli prefers to break down glucose, as we've talked about. In the absence of glucose, cyclic AMP, or CAMP as it's also called, will accumulate. So this molecule that you see in this picture will accumulate. CAMP has only one phosphate group, and it attaches to the ribose at 2 locations, which we've just explained. So there's something called CRP, and CRP is simply CAMP receptor protein. So this is found in the cytoplasm of cells. When CAMP binds to CRP, the complex together attaches to the CRP binding site, which you can see is right here. And it's better seen in this one right here. Here it's actually got the complex bound to it. So the complex, what happens is cyclic AMP will bind to the inactive CRP, and it will make this whole thing active. So this complex is now active, and it will plug into this active site. And what happens is it causes the DNA to bend. It causes the DNA to bend. This increases the affinity of the promoter to RNA polymerase. So the RNA polymerase has an increased affinity for the lac promoter. So the affinity of RNA polymerase for the lac promoter is naturally low. The RNA polymerase will bind to the lac promoter, but it's kind of casual about it. If it's available, it'll bind. No big deal. It's in no rush to bind. So it's naturally got a low affinity, even when there is no repressor attached. Binding of the cyclic AMP CRP, this entire complex will increase, and the bending of the DNA will increase the rate of transcription. And it does that by basically inviting the RNA polymerase to bind and then do transcription. So this actually increases the rate of gene expression. That's why this is called a positive gene regulation. So when glucose is present, there's little CMP. So glucose is up, cyclic AMP is down, and the reverse is true as well. So CRP is inactive and lactose operon does not function maximally. It'll work, but it doesn't work at its maximum capacity. So CRP affects other operons when glucose is absent. So this encourages metabolism of lactose. So the presence of cyclic AMP and activating the CRP will actually increase transcription. And it provides a backup system when glucose is absent, so that we can get that lactose, or so these organisms can get that lactose pulled apart to get at the glucose. So to summarize both positive and negative gene regulatory summary, CRP is an example of a positive gene control. When the molecule is active, it promotes the activity of the operon. So CRP is part of a positive gene regulation, whereas active repressors actually shut down the activity of an operon, and thus the repressors are examples of negative gene control. So when we were talking about lac operons and trip operons, those are examples of negative gene regulation. So if we take these two in combination with each other, both the positive gene regulation and the negative gene regulation, the organism can then fine-tune its metabolism as it needs to survive. So that's it for prokaryotic gene regulation. Next time we'll be talking about eukaryotic gene regulation. If you have any questions, let me know.